Moscow State University, Lomonosov, Moscow State University. I am a physicist and a computer science person who trying to work with medicine, with the development of new drugs. So I come here to discuss some problems in docking programs in general and the goal which can be established and problems which, for my opinion, can be solved to increase efficiency of application of computer science for new drug development. The simple problem to find a new molecule which will bind with a given target protein. Now predictions are very bad. Working prediction not for some competition, tests, benchmarks, but real working. We work with some real new drugs development and we know the problems. So I would try to discuss all these problems. Why it is impossible to predict exactly best champions inhibitors for a given target protein, making only calculations.
Yes. So you are in the drug discovery then?
Yes, drug discovery. But I understand that all this, your conference also interconnected with...
They are interconnected, yes, I'm happy for that. That's good.
Well, I'm Ashton Rogers. I recently graduated last year with a PhD in neuroscience, and I was just explaining it's the first PhD in neuroscience for the University of the West Indies. Saying that now, we don't have much back at the university. A very small program. I did my program actually under anatomy, but my supervisor and I created the neuroscience option. So I was ecstatic when I got invited to the conference to actually meet some of the great scientists who I've been reading about and working to meet. But not only that, I'm trying to create the Neuroscience Center of Trinidad and Tobago. Being a third world country, we have very limited resources, and most surgeries and stuff, simple surgeries, we have to outsource. Patients have to travel abroad and come back, and it's a very costly thing. We have low awareness of neurological diseases. Not many people are of the understanding of what they are, and as a result, we have a lot of stigmatization against neuro diseases. So it's my hope and my dream to interconnect with everybody around the world, neuroscientists, neurologists, psychiatrists, to help me create the center to actually alleviate some of the problems that we have. And also I want to pioneer a neuroscience department at the university. So we are very limited, but hopefully I can take that to a new level. And it's indeed a pleasure to meet, how do you say his name? Kjell.
That's me, right? Yeah.
It is indeed a pleasure.
Yeah. Well, I guess it's a good opportunity for all of us to introduce ourselves, and thanks you for starting it. Russia. And I'm from Sweden and working on in neuroscience, and especially on receptors nowadays and communication, and especially been involved with the fact that the receptors do not only exist as monomers, but they also exist in various types of complexes, homoreceptor complexes or heteroreceptor complexes, and represent their novel targets for drug development. And it's very complex, of course, but it's a fascinating new world with these novel complexes, and we certainly will discuss how this has resulted in the meeting on Parkinson's disease, how these new complexes can be novel targets.
Thank you.
And your couple will come in a second.
Oh, it's okay. Thank you. Thank you very much.
Symptomatic treatment and for protective treatment. So it will be interesting to see how the audience will discuss these results and ideas. So that's what we are involved with mainly at the moment.
Then I'm continuous. I'm Barry from UPF, from Lausanne, Switzerland, and I have nothing to do with drug deliveries or Parkinson's disease or any biology-related things. Unfortunately, it seems. So I'm a different branch of this conference is from nanotechnology. I'm a physicist, solid-state physics background, and what we're working on is graphene and nano and heterostructures, partially to use them in spintronics. So that would be an idea to use the spin, so the magnetic moment of the electrons next to their charges. Another field is make
Yes.
is to make heterostructures good for light detections, so visible light and in really high efficiencies. And that's,
Are you experimentalist?
yes,
Yeah.
I'm doing experiment.
Yeah, leave it. Okay. Thank you so much.
Or mainly measurements?
Thank you.
So it's fundamental science, but so in part of the group we also try to build like proof of concept to devices to see if they work.
This is Sharma. I'm working at Uppsala University, and my main interest is neuroscience, looking for neuroprotection and neuroregeneration, and looking for different types of drugs. Recently, we started 10 years ago work with US Air Force Research Laboratory for nanomaterials, and there are two aspects. But these nanomaterials can produce neurotoxicity, and these nanomaterials are also used for drug delivery. So together with US Air Force Research Laboratory and European Office of Aerospace Research and Development and some Swedish Strategic Foundation, we are working that can we deliver the drug to the brain for the treatment of traumatic brain injury at the moment. Then we are also studying hyperthermic brain injury. This is heat stroke related model, spinal cord injury, and some of the neurodegenerative diseases. We also started some models for Alzheimer's disease. And then we try to develop drugs. But no single drug can have, beneficial effect. So we are working on a multimodal drug at the moment in last 3-4 years, cerebralizing. This is the mixture of different neurotropic factors and active peptide fragments. So we are doing labeling with nanomaterials, nanowire. We are working at University of Arkansas Fayetteville. He has a US patent of nanowire, titanium nanowire. Then we are also labeling with PLGA, this soft nanoparticles, and try to inject in animal models and see that whether the mode of nano delivery is important or the drug itself is important. So these are the aspects we are looking last 6 or 7 years. So I am here in nanotechnology conference, and probably I am giving the first talk because the first person did not come. He was on water technology, so I am giving a talk at 9 a.m. as I was told. So this is my basic area of research. This is my wife. We both stay in Uppsala.
Okay, so I guess it's my turn. Anyway, my name is Shashi Paul. I'm head of one of the center in Montfort University, which is in Leicester. I think, I don't know how many of you have heard about Leicester city. It's not very far from London. It takes approximately 2 hours to drive. We were famous a couple of months ago because Richard III, the king, his skeleton was found under one of the County Council's car park. So I think that is one example. And we were very famous team rugby for Tigers. So I think, anyway, to cut short story, cut long story short, yes, I did my first degree BTech in electronics communications, and then PhD into condensed matter physics. I just want to say a few words that I think 20th century we spent to create millions of disciplines, physics, chemistry, communication. I think 21st century is aimed to combine those disciplines, and nanotechnology, I guess, is one of them, in which neuroscientists, material scientists, condensed matter physics, chemists, they all work together. Personally, I think whether you can think or not, that will end up into nanotechnology. So the center has a couple of disciplines. One is exploitation of nanomaterials into electronic devices, which I lead. Another, my colleague, Dr. Richard Cross, who works on the interface of nano and bio, especially understanding Alzheimer's disease and all that kind of thing. So he did. And young son, he leads into surface science exploitations of that. So I'm here to give a talk on exploitation of silicon into electronic devices. But you can say that silicon is already exploited into. But I think another area which has become very important as a material scientist. Materials are fabricated very or synthesized at very high temperature, especially the silicon. So it's like thousands of degree temperature. So when you heat these furnaces and all those things, this dissipate into environment. So it is a kind of a global warming. You consume a lot of energy to produce this material. One of my objectives is that can you lower the consumption of energy. So my talk is centered on that. So if you are interested, on 30th, please attend. Okay.
Thank you. I'm Neil Goodwin. I'm from California. I represent a firm. My office is in New York, Champions Oncology. So I live near Squaw Valley, California, because my wife wants to live at a ski resort, and I fly to work, and my office is in New York. We do tumor biology, so I'm a cancer pharmacologist. And so historically, drug development has used cell line models that then were xenografted into mice and drug compounds tested. And because the cell lines lost much of their human characteristics, the cancer drug discovery success is about 4%. And so about 6 years ago, we started cloning real people's tumors directly into mice from real patients and doing therapeutic testing on those tumors. And then to do that by the thousands. And so everybody's cancer can be, from a mutation standpoint, different. And so it gives you a personalized medicine modeling capability in biology. And second, we align with major clinical trial centers like MD Anderson, and we real-time make models by taking tumors out of real patients on clinical trials, putting them in the mice, testing the drugs, and then the mice inform which patient should go on which trial. So we're hopeful in the next 3 to 5 years of taking a phase 2 cancer trial success rate, which averages 10 to 15% of patients, to 70% by just more personalizing the decision making on who goes on what drug.
Very good. Yeah, we have a good team here.